Hello, I am Siwar Al-Ajloni. Our article today represents the modeling the activity of dopamine signaling pathway by combination of analog electrical circuit and mathematical approaches. In our model today, we have some agenda to talk about. What is the dopamine? Dopamine is a neurotransmitter that is produced in your brain in substantia nigra, ventral region, and hypothalamus. In this model, we studied the expected effect of changes in the functioning of calcium channels and mathematical model for the interaction between the components of the system. Today, there are many approaches to describe cellular process at the genetic level. This allows us to extend the idea of considering single complex biological system to many subsystems. For example, the analog between biological and synthetic electrical circuits, like the process of electrical circuit element operation simulate natural process of regulation in the cell, and the flow of electrons in transistors similar to the dynamic of biochemical reaction. The dopaminergic signaling pathway was chosen as the object of the study today. So, what is the DARPP32? Dopamine is responsible for triggering some cellular response. For example, a change in the conduction of calcium channels in nerve cells. In this figure, I will talk how DARPP32 phosphorylation can be controlled. Pathway of dopaminergic signaling phosphorylation is involved in the regulation and management of dopamine-induced transsynaptic effects. This is made possible by the association of D1 receptor with the activation of dinyl cyclase, which leads to the induction of CAMP synthesis. DARPP32 phosphorylation can be controlled by synaptic release of dopamine.
Hi, I'm Hala Mohammed Alaouni and I will discuss with you the functioning of DARPP32. The DARPP32 phosphoprotein, an important biological role in dopamine-sensitive neurons. Recent combination of experimental and modeling studies suggest that DARPP32 is a robust integrator of signaling, whose main role may be to increase the reliability in decoding the information mediated by dopamine, as well as other inputs such as glutamate. It is localized mostly to regions that receive dopaminergic innervation and present in virtually all medium-sized spiny neurons of the striatum. As shown in this figure, explain to us how the controlled and work of DARPP32. In this paper, we present the application of the hybrid approach for modeling of intracellular signaling pathways based on the combination of analog electrical circuit and mathematical models. And we will be focused on the dynamics of calcium channel current. The dynamics of calcium channel current behavior when the concentration of dopamine is changing during the signaling cascade induction by the activation of dopamine receptors of type D1 is considered.
Hello, my name is Anwar. Materials and method. Analog circuit for modeling, transcription, and translation of DARPP32. The use of transistor circuit allows us to determine the transfer function of the entire circuit. This task also implies the solution of the Mikhail-Menten equation ODE of the first and second order. II and D ISUP are the current proportional to the concentration of activators and repressors of the transcription of the protein of interest. XT is the total concentration of the transcription factor, DT is the maximum concentration of the protein, and KD is dissociation constant. Now we will talk about mathematical model. The effect of dopamine exposure and accordingly activation of the signaling pathway, change in the properties of ion channels, their conductivity. So pKa, which work is modulated by DARPP32, enhance the work of the CAV channels at highest concentration of ions calcium, weakening the connection of the coil medium with the terminal of the C1 channel. This conclusion was made by scientists. We are presenting mathematical model of the calcium channel when changing the concentration of dopamine. Here we can calculate the concentration of DARPP to 32 by this equation. To calculate the concentration of DARPP to 32 produced in a cell at some point over time, it is necessary to understand how the concentration of activators and repressors of particular protein transcription change in the cell with time. Of the resulting circuit voltage, which is proportional to the concentration translated DARPP to 32, depends on this equation. The calcium current is defined as Hodgkin-Huckley model. This equation was solved in conjunction with the control for the concentration calcium, like this equation.
So, this equation was solved in the conjunction with the control for the concentration of calcium.
For calculation and estimation, we use initial conditions for variables of mathematical model, like dopamine, protein phosphate 1, cyclic adenosine monophosphate, protein kinase.
Hello, I am Abdurrahman Abu-saleh and I will discuss with you section 3, results. System 2, solutions for different delta R can be either periodic, 0.5, 1, or damped 1.5, as shown. The frequency of oscillation of the activator and repressor concentration depends on the repressor decay parameter delta R, decreasing in time. Also noticeable is the change in the nature of the concentration dependence of this parameter. With its increase, the periodic dependence is replaced by a decaying periodic, and then a decaying non-periodic one. The frequency for the case with delta R equal 0.5 is 0.09 Hz. For delta R equal 1, it is 0.14 Hz. And for delta R equal 1.5, there are no oscillations. Such behavior suggests the idea of studying the joint dynamics of the concentration of activator and repressor DARPP32 over time. These signals show the trajectories of solution in phase space, which indicate the stability solutions of system 2. The obtained data allow us to say that, with an increase in the parameter delta R corresponding to the degradation of the repressor, the system tends to asymptotic stability. The profile of phase portrait evolution over time is presented on this figure, where critical value of repressor decay parameter corresponds to drastic change in external part of the profile's envelope. And here, this figure shows change in the phase portrait of system 2 as the repressor decay parameter delta R changed from 0.5 to 1.5.
This resulting shows us the change in the concentration of DARPP32 phosphorylated at residues of 309, 34, and 75. Change in the concentration of phosphorylated DARPP32 over time. A Vact minus Vrep equal constant. And for different repressor decay parameters, omega R, P omega R equal 0.5, C omega R equal 1, and D omega R equal 1.5. And in these signals shows us the dependence of calcium channels current in the presence and absence of dopamine over time, were obtained as a result of a complete mathematical modeling where concentration of DARPP32 is calculated using only equations 1 and 2 for blue line, and combination of modeling and experiment where concentration of DARPP32 is calculated using data from circuit and equation 1 and 2 in red line.
First of all, I am Yusuf Haiba, and today I'm going to talk about point number 4, the discussions. At first, there is a significant correlation between the calcium current dependency found in the simulation and the experiment. To look how the dependency were found, the coefficient of the determination in table 2 was determined. Based on the calculated values, there is a relationship between the result obtained as a result of modeling and experimenting with an electric circuit. The literature reports that the frequency of these oscillations can range from a few tenths of hertz to several hertz. According to the model, a change in the ratio of activator concentration causes a change in the frequencies of corresponding current oscillations. In the end, Further research will be aimed at studying the dynamic of biosensing of all proteins involved in the signaling pathway and assessing its effect on calcium channels work. Point number 5. Conclusion. Overall, the result demonstrate the capability of analog circuit approach via cytomorphic transistor-based circuit equivalent to simulate activity of such biological object as signaling pathways with a fair degree of accuracy. By using this tool, scientists can analyze the robust response of a complex network of reactions to an input signals as well as model some biochemical reactions and more realistic simulations that take into account biological molecular noise. We can draw the conclusion that the model presented in the paper, which is based on a combination of cytomorphic analog electronics circuits and mathematical approaches, can show satisfactory results with good correlation with previously published in literature data. This based on the good correlation with known experimental data.